So now we're going to talk about DNA profiling and what that actually means or entails. You guys might have seen on television such programs that talk about using evidence and DNA evidence, but you have to remember that most of our DNA is almost identical to each other. When you look around the room, you look at the diversity of the people, and there's only a small fraction of DNA base changes that are going to cause these great differences in phenotypes. So if that's the case, how do we determine who is who based on DNA? If all of our DNA is about 99.9% similar to the person next to us, we have to find sequences that are in place that will differentiate us. So we have to remember that forensics is using DNA profiling, or what they call DNA fingerprinting, in order to do this. Profiling depends on the variation in the genomes between person to person. Now, as I said, very few changes exist between us, but you have to remember that not all of our genome, all those A's, T's, C's, and G's, correspond to functional genes. A lot of it is spacer regions that are not necessarily going to have a direct influence on physical traits that you and I know. So we have different types of variations on genomes. One of them is called a single nucleotide polymorphism, or SNP, SNP, right? We pronounce it SNPs. And SNPs are very important because every once in a while you will have a nucleotide change that doesn't correspond to a change in genotype or phenotype, actually. Of course, it translates into a difference in genotype. Variability can also come from repeated segments, right? So when you have a tandem repeat, that means that you have these repeated elements right next to each other. As you can imagine, when a DNA polymerase is going to copy a genome, every once in a while there are repetitive units. If you looked at a sentence that had a repeating motif of similar words, how many times do you actually see those words? You lose count after a while, and the polymerase slips around and does the same thing. We have what are called VNTRs and STRs. VNTRs are called variable number tandem repeats. These are, as the name implies, repeated elements of nucleotides right next to each other, and they're variable. You can have one or two or three or five of these repeats. Likewise, STR stands for short tandem repeats. There's no real differentiation except for short tandem repeats can be much shorter, but there's no clear definition. So we'll use these words interchangeably. In the olden days, we had this technique called restriction fragment length polymorphism, and those of you who have taken the general biology class with us would have seen this technology in effect, where you were looking at the differences in pieces of DNA based on the way that they were digested by a specific restriction enzyme or restriction enzymes, as might be the case. This is poor technology because it requires a lot of DNA. Oftentimes, when you go to a crime scene, you don't see a big chunk of someone's tissue laying around. You only get a stray cell or so. So this is kind of hard to do unless you have a lot of DNA. Also, this requires a probe that will hybridize in a complementary fashion to a piece of known DNA. And this will give us different sizes on a gel or X-ray film, as might be the case, to indicate a pattern of these fragments, right? So each person will have a different RFLP profile if you're using a handful of enzymes, maybe usually two or three to be definitive. Now, the VNTRs or STRs, they're sometimes called microsatellites, and these are performed using PCR. And we will go over PCR in a little bit again, but PCR is an amplification technology, right? So therefore, if you have one cell, you actually have enough material to generate a profile based on the amplification of the DNA from this one cell. Therefore, it requires very little DNA. And because it's primer-based, it's very directed. You have to remember that PCR requires primers to direct where the replication or duplication of DNA occurs. Another word or name for this is called AFLP, or Amplified Fragment Length Polymorphism. Notice how it's very similar sounding to the RFLP or RIFLIP, right? But in this case, you need very little DNA, and you can watch this YouTube video to find out more about it below. Going back to what a RIFLIP is, or RFLP, you can imagine that in this case, you have these two alleles. Remember, we have two alleles because we're diploid organisms, and therefore we inherit one chromosome from mom and one chromosome from dad of each type. And here we have two of these alleles. They're called capital A and lowercase a. In this case, there really is no dominant or recessive allele, as they don't give rise to a phenotype that you actually see from expression. So therefore, the trait itself is a phenotype, the actual sequence. As we can see in the capital A, there are these three diamonds that correspond to the restriction site of some sort. So you have some sort of enzyme that will cut at 1, 2, or 3. However, in the lowercase a, there are only two of these. Notice that position 2 is missing in this case. When we look at this location here that has this thick rectangular boxed area, that corresponds to the area where the probe will attach. So when we look at these two different alleles, you would get this intermediate size, or this long size piece of DNA that will show up by separating on an agarose electrophoresis gel, whereas the capital A will generate two fragments, right? One here and one here. We're not interested in the stuff outside, but the probe will only bind here. So therefore, you will get a short piece. And that's how we would differentiate these two alleles. Now imagine a restriction site would exist on many pieces of DNA because we have so much DNA. Therefore, it will it'll generate a lot of fragments, but only a few of them will be specific to this type of probe or sequence that we have there. How did this restriction site 2 come to an existence? Well, it could be an SNP, right? We don't know which one came first, but an SNP here would abolish a restriction site, or an SNP here would generate one. We don't know. All we're interested in seeing is that they are, in fact, there or not, right? Now, you can also have these two alleles called C and D. Remember, both of these just correspond to, there is no dominant or recessive, but if you look at this, the C has these little boxes here. And they also have this area where the probe is going to bind this dark rectangular region that is in both C and D. And if you look at this, we have 1, 2, 3, 4, 5 of these boxes in allele C. Whereas in allele D, we only have 2 of those boxes. Notice that the restriction sites are in the exact same location in this case. Now, because we have this element that has been repeated and differentially expanded, then we have a different number of tandem repeats. And because when we have these different repeats and when we digest this pieces of DNA with our enzyme of interest, and we hybridize our probe to this dark rectangular area, this one automatically will be larger because there are more repeated elements in there than D. So repeated elements are just expansions of, say, a few nucleotides or so of the same repeats. Here we can look at what these patterns look like in an agarose gel. And when we think about it, we can look at parenting and sibling relationships using this. Now, if you took all of your DNA and digested it with a restriction enzyme and you ran out of gel, you would see a big smear. You wouldn't be able to differentiate any of the DNA, right? But you would use a probe specific to these spots that we see here or here. And by using those probes with radioactive DNA, you can actually see individual bands at which this unique profile exists. So as I said, there is no dominant or recessive because they're both expressed. However, they don't correspond to a physical trait that you see, just a sequence. So if we look at the genotypes here, capital A generates the smaller one, as we see, because it's the fragment between 2 and 3. And lowercase a generates a larger fragment because they don't have this position 2. Notice that you only see one band in each, even though each person is diploid. That's because if they're the same size, you can't distinguish them. The heterozygous condition here is if we have one capital A and one lowercase a in an individual, and therefore you can see what that looks like. You will see 2 bands. So if we have 2 heterozygous parents, we would have this type of profile. And we can see that it's possible for these heterozygous parents to generate 3 offspring with these different alleles, right? And we have to remember that the heterozygous condition is going to be in half of the offspring, more often than not, just based on statistics. Remember basic genetics, when you do your Punnett square, it's done the same way. Now, remember what I said. Oftentimes, you're dealing with billions of bases. This is just some random sequence. So how do you figure out the significance or differentiate people based on all of this DNA, especially when you're dealing with such a tiny amount? Well, remember we talked about how PCR works. You have a DNA polymerase, and a DNA polymerase always works in a 5' to 3' fashion. However, a DNA polymerase can't start from nothing. It always starts from an RNA primer in nature, right? The RNA primer is put there that's complementary to a piece of DNA, and it is here in red. That corresponds to the RNA primer that will just get it started. Remember what priming means. It means to get it started. And once that happens, the DNA polymerase will work on its merry way in a 5' to 3' fashion. But at each replication bubble, there are 2 strands. So then there's going to be these other strands here that are discontinuously made because this end hasn't opened up enough to allow priming and an extension event to occur on the template, right? So all this requires in nature is an enzyme that will unspool the DNA. It's called helicase, right? And there's a primase that will add a primer in red. And you would have DNA polymerase that would just go and extend this. Now, remember, each replication bubble has 2 of these forks here. So on the other side, there's going to be the mirror image of this, right? And we call this strand here, the leading strand because it's going to be continuously made, whereas this is referred to as the lagging strand because it's discontinuously made. It has to catch up, and the only way it can catch up in being made is every time this helicase unwinds a new stretch of DNA, a new piece can be generated. Remember, it's semi-conservative. And these fragments on the discontinuous strand or the lagging strand are referred to as Okazaki fragments. So in a tube, we often use an enzyme called DNA polymerase 1. We don't use the whole thing. We actually use this portion called clean-out fragment. And the clean-out fragment has this area removed from that protein because this area of that protein is responsible for removing the primer. And if you're going to do it in a tube, you don't use an RNA primer. You just use a DNA primer. So you don't want that to be chewed up or removed. You want it to stay there. And this is what we've been using for many, many years in a tube to generate exactly 1 copy of DNA. What you do is you have to separate the 2 strands. We don't have helicase, so we do this by using heat to denature the 2 strands of DNA. Then you cool the solution after you've added a primer that's in abundance. It might have been there already. And what happens is when there's this DNA primer in abundance, it outcompetes the original strand of DNA because there's so much of it that it will hybridize there. Then you add your clean-out and your DNTPs, and the clean-out fragment is this DNA polymerase that will just chug along, filling in that space to make a copy. Of course, you need to do this at a physiological temperature, like 37 degrees Celsius, because that's what E. coli like. And as I said, you make just 1 copy. That's not a whole lot. So how do I make multiple copies in a tube? Well, in the olden days, you would just take the same clean-out fragment, and as we said, you first denature the DNA because you don't have helicase. It splits the DNA apart into its 2 strands. you have a lot of primer in the solution so that when you cool it down, it hybridizes to these primers. Then you add your cleanout fragment with your DNTPs floating around and the appropriate salts and buffers. And what will occur after you put at 37 degrees is an extension of this DNA from the primer. But then if you want to make another copy, you'd have to denature it again. And you'd have to denature it by putting it back at 95 degrees Celsius. However, we know that if you put proteins at 95 degrees Celsius, that tends to cook it or clean or denature it as well, and therefore it will kill it and it will not be functional anymore. So we end up cooling it back to a temperature that is amenable, and by cool, it's a relative term, like around 60 or 50 degrees Celsius, which is very hot still. And remember, there was an abundance of these primers, so they would have reannealed. And at this point, all you would do is add more enzyme, right? And then you would go back to step one. And every time you go back to step one, that would kill the enzyme. That's the only way that you can separate the two strands of DNA, with heat, because you don't have helicase constantly active, right? And you do this about 25 times or so, and you make many, many copies. However, this is very tedious and uses up a lot of cleanout enzyme, which is kind of expensive, because making pure proteins is an expensive endeavor. And it requires a lot of attention. Someone actually had to stand there, and we're talking about orders of magnitude of like, a few minutes for each cycle. So therefore, someone had to actually change this tube from one heat block at these different temperatures to another, and then put them on ice and add more enzyme. So what if we didn't have to keep on replenishing that enzyme? Well, in 1960s, a DNA polymerase was isolated from a bacterium called Thermus aquaticus, and this lived around geysers and hot springs. So therefore, the temperature is a little bit higher to begin with. They found that Thermus aquaticus thrived in high temperatures, about 70 degrees Celsius. And this type of temperature, by the way, if you touch something at 70 degrees Celsius, it will hurt because it'll start to burn you. And many of the proteins in this bacterium could resist denaturing temperatures, including the DNA polymerase, because it lives in that environment. It's adapted. The Thermus aquaticus usually lives in a range from 55 to 80 degrees Celsius, which is hot. The enzyme is capable of resisting 95 degrees Celsius without being denatured. So that makes it ideal for using in this type of situation. In 1983, a man named Kary Mullis got tired of constantly replacing and moving these tubes from different heat blocks and buckets of ice. So what he did was he read about this enzyme and decided if he used this, he wouldn't have to replace that cleanout every single time. And we now call this DNA polymerase TAC, for Thermus aquaticus polymerase. And in 1993, he won the Nobel Prize for automating this system. So this was called polymerase chain reaction because it's a chain reaction of DNA replicating DNA. And it's outlined to the left, as we see, where actually every cycle of denaturation, annealing, elongation ends up doubling the amount of DNA that is present of a specific flanked region, right? Remember, the primers will flank a region of interest. So you're not copying the entire genome of something. You're just copying specific areas. And based on how long you let it elongate, and based on how well the polymerase hangs onto the DNA, will dictate how long this piece of DNA is based on how flanked, where the flanking occurs, right? Otherwise, if you let it go on forever, it'll just make one copy and fall off eventually, but not make a whole copy. PCR enables for the rapid and automated amplification. As I said, each time is a doubling if it was perfect, so therefore it's exponential. Using the thermostable enzyme means that you make one mixture, you set it and forget it. You put it on a machine that cycles between different temperatures, and it goes to work. As we said before, you do it 25 cycles, 30 cycles, and it will make many, many exponential copies. You can follow the links along to below to go visualize this in a different way, those are very useful resources there from the DNA Learning Center. Now, going back to this variable number tandem repeat business, that's what we were trying to get at to begin with. It was discovered in 1984 by Sir Alec Jeffries. He was the first person to actually use this, what we call junk DNA, the stuff that's between genes, or intragenic DNA. It doesn't code for any of the protein, right? You have to remember a very small amount of our genome corresponds to proteins that are transcribed. We have a lot of pseudogenes, right, in there, which also counts as junk because they're not real genes, or they used to be genes that ended up being mutated out of existence, or they may be regulatory elements like microRNAs. VNTRs or STRs are fairly short, as we said before. We'll say that they're between 3 to 30 base pairs, and they could be repeated multiple times, 10, 20, 100 times. We have an example here of CAT, 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 which is just an easy one to remember because it's just 3 long. So what is variable is the number of these copies. So if we look at the example, one allele might have 3 copies, cat, 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 and another allele might have 5, cat, 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 right? So we can see that there's a clear difference in the length of this. That's what we mean by tandem repeats. Here we have an instance of 2 individuals, a mother and a father. And as you can see, mom here has an allele on one chromosome that has 3 at this location. At the other chromosome, she has 8 of these repeats. Whereas dad has 5 on one chromosome at that location, and at the same location on the other homologous chromosome, he has 7. So the child can have a possible VNTR allele from these 2 individuals, as we see here. You do a simple Punnett square, and you will get these results. As we said before, that this was an amplification-based technology. So it requires you to know a sequence to prime from. And the primers, as we see in figure A here, are always outside, just outside of where the repeated elements are. So we know that area, right? We know the sequence of those areas that are flanking these repeated regions. So what we can do is amplify these specific regions. Now, remember, just as many people will have brown eyes, you can imagine that people will have the same number of repeats in their genome. So what we tend to do is not just look at one VNTR. We tend to look at a handful, 3. But in forensics, we'll actually look at 13 because that's going to be very definitive, right? And you can see in this multiplex PCR where we have actually 3 different STR locations or loci, and they're being amplified using their specific primer sets. And imagine that they'll give rise to a different color product. This is almost entirely true because what we'll often do is we'll change those primers, we'll modify those primers so that they're fluorescent in a different wavelength, right? And therefore you can see multiple colored products at the same time. This is how you differentiate them. Because what if you have 2 STRs that are, even though they're from different loci, they're very similar in size. You have to differentiate them in some way. And when you multiplex them, you can see what happens when you multiply them and replicate them and run them on this gel. You have this specific pattern. Notice that these individuals don't have the same pattern of these 3. So we can differentiate them. And actually, that's not entirely true because in this case, individual B and forensic sample F are the same, which implicates individual B in whatever happened, either a crime or a paternity case, etc. Right? We can see that B and F have the same profile of banding. So the repeat sequences in DNA are very important because they're not selected against. When they're outside of genes, they're free to mutate because things that are inside of genes tend to be very important and you wouldn't want them to mutate. So chromosomes within a species vary in length, just by a little, nothing noticeable. And these stretches of repeated DNA, as we said, are outside of genes. But sometimes we have these repeated DNA sequences inside genes, and as we can see, they cause diseases, such as found in Huntington's disease, which is a repeated sequence of CAG within the Huntington protein gene. We also have these CAG repeats inside of other diseases related to Huntington's disease, and they do correspond to proteins that are expanded as well. These stretches are found tandemly or right next to each other, and as we said before, they're called VNTRs, STRs, or microsatellites. Because we're using PCR, it means there's very little DNA needed. Using primers flanking outside of these regions make it very specific to those loci. And the amplification, based on whatever the repeat number is, will differ in size. Here's one that we don't use in forensics much. It's called the D1S80 locus. And as you can see, in each one of these wells, corresponding to a different individual, right? This is one person, this is another person, that's another person. Notice that they have two of each band. That's because they have two different alleles, one from each parent, right? This one is not used so commonly in forensics because there is a bias towards certain ethnicities that have this very similar pattern. So this one's not used very often, but we may use it in class. TH01 is a name of one of these loci of another STR region. And we happen to know that inside this locus, there is this repeat of TCAT, or we'll call it TCAT, right? So we're only interested in how many times this TCAT sequence is repeated. We're not interested in the sequence itself, right? We just happen to know that it's a stutter, if you want to call it that, of TCAT, TCAT, TCAT. So we have primers outside of this that we know exist at this location on the chromosome. And we can specifically prime and amplify this region. Here we can see that an individual has two TCAT sequences on one chromosome, and on the other chromosome that is homologous at the same exact location, we have 4. Remember, we're diploid, so that we have one from each parent. And we can use this in such a way to identify paternity, maternity, or forensics, right? The physical number of these repeats is the trait. There is nothing more to this. If we amplify this region using the primers that exist outside of this region, we will amplify this repeated sequence. And when we run it on gel, it will look like this below. Our PCR sample will have a larger band of 4 and a smaller band of 2. And we can see that if we amplify someone suspected of being the father, has one around 2 and one around 5. And if we look at the PCR of the suspected mother, usually you know who your mother is, then she also has a band at 4 repeats and one at 3 repeats. So this pair of parents probably are the parents. Okay, it's not definite because, like I said, many people have brown eyes. It doesn't mean anything. They might have brown eyes and curly hair, or brown eyes and straight hair. That doesn't mean that you're the offspring of people with those attributes. That's why we do multiple VNTRs or STRs when we're doing forensics or paternity. In crime, we have this system called the Combined DNA Index System, or CODIS. And CODIS is very important because it's developed by the federal government with local agencies involved as well, and they use this to have an index of criminals, right? They use 13 different markers, and one of these markers is called AMEL. AMEL is on the X and the Y chromosome. Right? Remember that these two are not homologous. However, we can tell that the AMEL locus on this is a different size than on the Y. So you can use it to detect gender. We use the same markers to detect not just criminality, but also paternity and maternity. And you can see that there are all of these different funny names that correspond to a locus. And these locus have specific primers that will amplify those regions of interest to help us identify relatedness. Uh, in 2013, there was this article here or news item that said a woman's DNA found on Boston bomb. And the reason why they knew there was a woman who touched the bomb, not to say that she was implicated in any way, is that they simply used CODIS markers and they were able to identify just the presence of the X, AMEL locus. If there was a man involved, which there was, obviously, they found DNA that had two different alleles, right? So this is just an example of how CODIS works, how forensics works, and how we've come to use amplification to make sure that we can have a lot of data or evidence from very little tiny pieces of DNA that lay around from either hair or urine or other types of bodily fluids at a scene.